dann würde ich Professor Pascolo einladen, noch seine Ausführungen darzulegen. Herr Professor Pascolo, Sie sind seit 20 Jahren in der Forschung der mRNA-Technologie tätig und Mitbegründer der Firma CureVac. Können Sie uns die mRNA-Impfung und ihre Funktionsweise erläutern und vielleicht eine Antwort geben auf die Frage, wie gut schützt denn die Impfung vor dem Virus und was sind denn die Risiken, wenn es welche gibt und was ist der genaue Unterschied zu den Vektorimpfstoffen?
Thank you for the invitation. Sorry, I will switch to English. I hope it's okay. Do you hear me well?
Yes, it's okay.
Do you see my screen?
Yes, we see your screen.
So let's start. Yes, so there were a few questions about the mechanism of action of RNA vaccines and the history of RNA vaccines and the safety of RNA or DNA vaccines. So I hope that I can, in the next 14 slides, clarify these points. Let me first remind you the central dogma of molecular biology, telling us that there is a flow of information from DNA to messenger RNA and then to proteins. In rare occasions, the messenger RNA can be reverse transcribed into DNA, but this is very specific of messenger RNA from retroviruses. DNA is double-stranded and consists of 4 bases, SCGT. The genes in the DNA are transcribed into messenger RNA. Messenger RNA consists of 4 bases as well, ACGU, and then the messenger RNA is translated into a protein. There are 20 amino acids that can build proteins. DNA is very stable, it remains. On the contrary, mRNA and proteins are transient, they are constantly produced and degraded, so that the cells have a dynamic that's due to the fact that mRNA and proteins are always produced and destroyed while DNA is stable. In the eukaryotic cells, the cells that we have, us, mammals and humans, the DNA is separated from the rest of the cell. It's in the nucleus, and then in the nucleus, the DNA is transcribed into messenger RNA, and the messenger RNA goes out of the nucleus through pores, and then in the cytoplasm, it's being translated. If we look at the vaccines we had until 2019, it's a non-exhaustive list here with in color the vaccines that are suggested in Switzerland. I group them into, actually in 3 groups: attenuated viruses, inactivated dead viruses, and subunit vaccines. The inactivated viruses and subunit vaccines are dead vaccines, so whatever is injected cannot live. On the contrary, the attenuated viruses are live vaccines. And here let me point to the fact that some of these attenuated viruses contain messenger RNA as genome, and some contain DNA. So keep in mind that these old vaccines, they are also messenger RNA or DNA vaccines. They are viruses. When they are injected as vaccines, the people who are vaccinated take the mRNA or the DNA from these viruses, make the protein of the virus, and trigger an immune response. It's not the protein in these viruses that are contained in the syringe that trigger the immune response. Is the genetic information of these viruses in form of messenger RNA or DNA that is taken up by our cells, and this genetic information is expressed by our cells, which trigger then the immune response. If I put the different vaccines into the central dogma, again, you can see that the DNA viruses, the live DNA viruses that are used as vaccines against smallpox or chickenpox, can be considered as DNA vaccines. The live vaccines that protect against yellow fever, mumps, measles, rubella, they all are mRNA viruses, so they can be also considered as mRNA vaccines. Now, a few newer vaccines, like hepatitis B, human papillomaviruses vaccines, they are recombinant proteins. The influenza vaccine, the vaccine against grippe, can also be considered as a protein vaccine because it's an inactivated virus. So it's the protein content in this inactivated virus that is used for the immune response. In the context of COVID, the same formats have been used to develop vaccines, DNA, RNA, protein. DNA vaccines are the AstraZeneca, Sputnik V, Janssen vaccines. They are adenoviruses, so they are replication-deficient viruses. They can still be categorized as live vaccines because they are viruses that infect. They are DNA viruses that infect, so it's the DNA that is transferred from this virus to your cells when you are vaccinated, and this DNA is taken up, is transcribed in mRNA, and then the mRNA is translated into protein to give an immune response. So these three vaccines are actually DNA vaccines. Then there are the mRNA vaccine from BioNTech, developed with Pfizer and Moderna, and there are a few protein vaccines developed against COVID-19, like the Novavax vaccine. And Switzerland has reserved from AstraZeneca, Janssen, BioNTech, Pfizer, Moderna, and Novavax. The production of synthetic mRNA vaccines is described here. It's very easy, and that's why it was so fast in 2020 for the mRNA companies to develop this mRNA vaccine, because the production in vitro of messenger RNA is very easy. It consists of having a few reagents. First, a DNA of interest. This is the DNA of interest. You can buy it. You can buy DNA coding for spike protein of SARS-CoV-2, or for protein of influenza, or for protein of cancer, any protein of interest. And then this DNA has a sequence that controls the recognition by a certain RNA polymerase that you also buy, recombinant RNA polymerase that is made by bacteria. And so you just mix your DNA and the RNA polymerase together with nucleotides and some analogs, and within a few hours, the DNA in vitro will be transcribed into messenger RNA. From one molecule of DNA, the RNA polymerase can do more than 1,000 molecules of mRNA. It's like a photocopy machine. The DNA is the book, and the mRNA is the photocopy. So you put the book on the photocopy machine, you press 1,000, and you get 1,000 photocopies, independently of the page of the book, independently of the DNA of interest. So any DNA of interest will be transcribed into the messenger RNA. The process is robust. That's why it's so easy, again, to produce messenger RNA vaccine, because independent of the mRNA that you want, independent of the DNA that is here, you will always get your mRNA as simple as doing photocopy from any page of one book. So after a few hours, you get your plenty of messenger RNA. You put a DNase, an enzyme that degrades DNA, and then you can just purify the messenger RNA, for example, by precipitation or by more advanced purification methods to have pure messenger RNA. Again, all these steps are robust, so they work whatever mRNA you produce, coding for whatever protein you want to use in the lab or in the clinic. Once you have a purified messenger RNA, you need to formulate it in order to administer it. And here is a drawing of the mRNA vaccines from Moderna or from BioNTech, Pfizer. So it's synthetic messenger RNA into a liposome. The liposome consists of 4 lipids that make then vesicles that are less than 100 nanometer big. And these vesicles, they protect the messenger RNA and they deliver it to the cells, like the mumps, measles, yellow fever viruses deliver their mRNA in your cells when you get vaccinated. Here is just an example of the results obtained by published by BioNTech to show how strong are these mRNA vaccines. These are in the sera of volunteers injected by the vaccine. The titers, the neutralizing titers against SARS-CoV-2 has been measured. They are placebo people. They receive placebo. They don't have in their serum any antibody that can neutralize SARS-CoV-2. Here on the other side, you have the sera from people who have recovered from COVID, from infection. You see they all have neutralizing antibodies against the virus. And here in the middle, you have younger people and older people injected with different doses of the vaccine. This was the phase 1 dose escalation study. And you see at day 28, so 1 week after the second injection, in all doses, 10 microgram, 20, 30, the people have high antibody titers that can neutralize the SARS-CoV-2, showing how good this vaccine is, also with 10 microgram in adults, 20, 30, and this is also the case in elderly people. Again, I want to stress that mRNA vaccines are not new. You had the feeling maybe that you got a new vaccine. It's true in sense of production, but in sense of mode of action, it's not true. Again, the yellow fever, mumps, measles, rubella viruses are messenger RNA viruses, and the vaccines containing these attenuated viruses, when they get injected to you subcutaneous without adjuvant, it's not the protein in the viruses that trigger the immune response, it's the messenger RNA that is actually transferred from the virus to your cells, and your cells from the messenger RNA produce the protein of the virus. It's the same mode of action now for this synthetic messenger RNA vaccine. So you receive these vesicles, and the messenger RNA contained in the vesicle go in your cells, and your cells produce the proteins of the virus, which allows to make an immune response. So it's the same mode of action for the new synthetic mRNA vaccines compared to the old natural mRNA vaccines. The yellow fever vaccine is nearly 100 years old. A few facts on synthetic messenger RNA vaccine. First, messenger RNA is very stable. You can store it at room temperature. Of course, when it's not contaminated with RNAs, which are enzymes that degrade RNA, and our body, our cells are full of RNAs. That's why the RNA in vivo is quickly destroyed. But in vitro, pure messenger RNA is very stable. What is not stable so far in the vaccines are the liposomes. These are bubbles of fat, and if you leave them at room temperature for a long time, they can aggregate, they can open and release the messenger RNA, or they can change in size or form over time. So that's why the vaccines of BioNTech and Mrna have to be stored frozen. It's because the liposome is not stable. The messenger RNA is very stable. It's just so far the formulation is not stable, and new formulations will come, which will allow us to make better and more stable mRNA vaccine formulations. The third point, which is important, is actually that the messenger RNA vaccines, the synthetic messenger RNA vaccines, are vegan. The production that I showed you on the previous slide uses bacteria from enzymes and chemicals like nucleotides. So the whole process does not involve any animal products. And this is important first for the environment, because it's less polluting. For example, when you need to produce the flu vaccine every year, the flu vaccine is produced in eggs. So you need millions of chickens to produce millions of eggs in order to produce the flu vaccine in the eggs. And here with the synthetic messenger RNA vaccine, you don't need any animal products. So it's less problems with the animals, it's less problem for the environment, and it's also less problems with allergies, because quite a lot of people have food allergy, egg allergy. They cannot get certain vaccines because they are allergic against eggs. And so if the viruses are produced in eggs, then people cannot get these vaccines. So this is not a problem with the synthetic mRNA vaccines because they are vegan. And then a fourth point, it's easy to produce the messenger RNA vaccine, and it also needs relatively small infrastructure. In 6 liters of the reaction in vitro transcription that I showed you on the previous slide, so the DNA together with the RNA polymerase and the nucleotides, in 6 liters of this reaction, in a few hours, you have enough mRNA for 1 million of doses of mRNA vaccine. If you want to compare, to do 1 million of doses of a virus in culture, you usually need 1,000 times bigger amount of production. So you compare it to 1,000 smaller production facilities that you need to produce mRNA vaccine compared to producing viruses. And of course, safety is important. When you produce viruses, you have to be sure that there will be no leak in your production and no contamination to outside. When you produce mRNA, it's not a problem. If mRNA would get out, it will be degraded by the RNases that are everywhere. A little bit of history on the messenger RNA, and I introduced here maybe because we're in Switzerland, it's important to remember that it's a Swiss doctor, Friedrich Mischer, that has discovered nucleic acids. He was born in Basel. He went then to Tübingen. in the 19th century to do research in the kitchen of a castle that was transformed in a lab. And if you go there now, you will see it has been transformed in a museum about molecular biology. So here in this castle, Friedrich Mischer has used blood cells and has extracted the nuclei of blood cells, and he has found in the nuclei of blood cells a new substance that is called nuclein. And after 2 years, he came back to Basel. He continued his research on the nuclein, and he could isolate the bases and go on. The research went on to then show the DNA, RNA, and the components. It took quite some years then later to discover the messenger RNA. It was discovered in Paris in the Pasteur Institute, mostly by François Jacob and Jacques Monod, who received the Nobel Prize in 1965 for their discovery of the messenger RNA. Here the drawings are made by Caroline Schubach. It took 30 years to use messenger RNA in a vaccine. And the first publication of messenger RNA vaccine is again in France, again in Paris. The team of Professor Melien has produced synthetic messenger RNA in a liposome to vaccinate mice against flu. So both the discovery of messenger RNA, this natural molecule that is in any of the cells that makes our body, to the first synthetic mRNA vaccine, this was all made in France, in Paris. Then the first injections of messenger RNA in humans, I made them. I got actually messenger RNA. I decided to get injections of messenger RNA coding for firefly luciferase. This protein is responsible for light production in firefly. We produce messenger RNA coding for firefly luciferase. I got injections of this synthetic mRNA in order to see whether the synthetic mRNA would be safe in human and whether it would be functional in human. And we could publish that in 2007, that synthetic mRNA works in human and is safe and expressed. And we did also the first clinical studies of messenger RNA vaccine in cancer patients, in melanoma patients, and in renal cell carcinoma patients. We published these clinical studies in 2008, 9, and 11, and we could show for the first time in human that synthetic mRNA is inducing an immune response in human. And during all these 20 years, there were the fantastic research of Ugo Sahin and Özlem Türeci in Mainz, and their work in the last 20 years has allowed BioNTech to validate the messenger RNA vaccine against COVID. During all those years, I started in 1998 and until 2010, the messenger RNA vaccines was the ugly duckling of the vaccine field. All the investors and researchers were focusing on DNA vaccines, protein vaccines, viruses vaccines. So there were big support for this kind of vaccines, not for the mRNA vaccines. There were some contempt actually against mRNA vaccines. And of course, this has totally changed. We had the pandemic, the SARS-CoV-2 pandemic in 2020. We don't know whether it comes from live animals in wet markets in China being eaten like this bat in the sandwich, or whether coronaviruses adapted in labs to grow in human cells could escape from a lab. But this pandemic has totally changed the view that the medical and scientific community has now on mRNA vaccine, and all the contempt that we had until 2019 against mRNA vaccine has now been transformed into a great interest. The whole world is now looking and developing and using mRNA vaccines. Here I show you a little bit of some of the ongoing clinical studies of synthetic mRNA vaccines against different viruses. Of course, SARS-CoV-2 has been done, and rabies, HIV, Zika, influenza is ongoing. So we can expect in the near future to have new synthetic mRNA vaccines, for example, against influenza, replacing the traditional vaccines which require eggs and growing viruses. So again, the synthetic mRNA vaccine is easier to produce, it's vegan, so it has only advantages. There are also a lot of mRNA vaccines formulation being tested against cancer, melanoma, ovarian cancer, prostate cancer, a lot of solid cancers. So we can expect also in the near future to have synthetic mRNA vaccines against cancer being validated. There is more to mRNA than vaccines. As I said, vaccines against all viruses are being developed, so we will replace probably the old vaccines, again, even yellow fever, mumps, etc., with a synthetic mRNA vaccine being easier to produce and safer and not giving any problems of allergies and the disadvantages I told you about. The mRNA vaccines against cancer are being developed, so mRNA codes for cancer protein, and can be completely individualized. It's the haute couture of vaccines where every patient gets its own mRNA vaccine coding for mutations that has been found in each patient's cancer cells. There are also development of mRNA vaccines against allergies. The contrary of vaccines is tolerizing the immune response, and this can also be achieved by certain formulation of messenger RNA given intravenous. So you can deactivate specifically the immune system with messenger RNA, and this is in preclinical studies and will soon go in clinical studies to treat patients having autoimmune disease like multiple sclerosis, where you can deactivate the autoimmune response thanks to messenger RNA vaccine or devaccinating formulations. Then there is a big use of messenger RNA, synthetic messenger RNA for therapies. So here we don't speak anymore about vaccines, we speak about therapies where the messenger RNA is used to express proteins like EPO, erythropoietin, or antibodies. There is also quite some research and clinical studies ongoing with regeneration. We are not yet at the stage where mRNA can completely regenerate people, but what can be done, and is in phase 2 clinical studies, is to use messenger RNA to grow new blood vessels at the surface of the heart after a heart infarct. This is in phase 2 by Mrna. So they inject mRNA coding for VEGF, vascular endothelial growth factor, which is a protein that allows to grow blood vessels. You can use messenger RNA to repair the retina, the skin, the muscles, to make neurons. You can reprogram cells, make stem cells, SC, or make anti-cancer T cells. So these are also really high focus of the synthetic messenger RNA research community. And you can also use synthetic messenger RNA to modify genomes, to correct genetic default by using messenger RNA coding, for example, for Cas9, a bacterial protein, which in some precise conditions can change the DNA in order to repair a genetic mutation, genetic disease. So with that, I finished. I'm happy to answer your questions if you have questions.
Thank you very much for this very insightful presentation.